Gluconeogenesis is the process of synthesizing glucose from non-carbohydrate precursors. The liver, and under extreme conditions of starvation, the kidneys, are major gluconeogenic tissues. Of note, the kidneys can contribute as much as 40% of total glucose from gluconeogenesis in the fasting state, and even more in starvation. Although the glucose synthesized by the kidneys under these circumstances are really intended for the renal medulla, some of this glucose find their way into the bloodstream. Recall from the previous video series that when glucose stores become depleted and blood glucose levels fall to low levels, glucokinase in the liver is inhibited on account of its high Km value. Since the liver is not highly dependent on glucose, it will not use the meager glucose supplies that remain available. In fact, the liver will even synthesize new glucose from non-carbohydrate precursors. This is important because cells such as neurons and red blood cells, which are critically dependent on glucose, can continue to scavenge blood glucose on account of their hexokinases, which have low Km values and thus high affinities for glucose. That way, the viability of the brain and red blood cells is protected. So, in the event that gluconeogenesis fails to occur, it is usually fatal. Conversely, excessive gluconeogenesis occurs in critically ill patients in response to injury and infection. This contributes to a state called hyperglycemia or high blood glucose levels, which is an independent predictor of mortality among these types of patients. A major source of carbons needed for gluconeogenesis is lactate produced by skeletal muscle and red blood cells. Another major source of carbons is glycerol, coming from the adipose tissue. Perhaps the most important of the major carbon sources for gluconeogenesis are glucogenic amino acids, particularly alanine in the liver and glutamine in the kidneys. Propionate is another carbon source for gluconeogenesis, albeit a minor one in humans. In ruminants such as cattle, propionate becomes the principal gluconeogenic substrate. The overall goal of gluconeogenesis is to basically maintain a supply of blood glucose necessary for the nervous system and red blood cells during times of fasting and starvation. After an overnight fast, gluconeogenesis, along with a process called glycogenolysis, which is breakdown of glycogen to its constituent glucose units, contribute just about equally to blood glucose. When glycogen stores become depleted, as what happens in starvation, gluconeogenesis becomes progressively more important. Recall that lactate is one of the major sources of carbons for gluconeogenesis. Under conditions that call for gluconeogenesis, lactate is oxidized to pyruvate via a reversible reaction catalyzed by lactate dehydrogenase, which is dependent on NAD+. Recall that this is the reverse of what happens during glycolysis under anaerobic conditions, when lactate is formed from the reduction of pyruvate. It is gluconeogenesis that clears the lactate produced by muscle and red blood cells. Glucogenic amino acids also get converted to pyruvate via a transamination reaction. Glucogenic amino acids are exemplified by adenine in the liver, wherein its amine group is transferred onto alpha-ketoglutarate, converting the latter to glutamate and the former to pyruvate. Recall that, depending on their metabolic fates, amino acids can be classified as either ketogenic amino acids, which yield acetyl-CoA or acetoacetyl-CoA, or glucogenic amino acids, which yield pyruvate or Krebs cycle intermediates. The latter are called that way because their carbon skeletons can be used to synthesize glucose through gluconeogenesis. On the other hand, the breakdown products of ketogenic amino acids, particularly acetoacetyl-CoA, can be converted to ketone bodies. Neither acetyl-CoA nor acetoacetyl-CoA can bring about net glucose production in humans. This is a table showing the distribution of amino acids according to this aforementioned classification. Notice that there are only two essential amino acids that are purely ketogenic, namely leucine and lysine. The glucogenic list is significantly longer, with 13 amino acids under this group. And there are also amino acids that are both glucogenic and ketogenic. Let's take tyrosine for example. When tyrosine is broken down, one molecule of fumarate and one molecule of acetoacetate is produced. Now, those amino acids in pink boxes are the glucogenic ones. But for our topic, gluconeogenesis, we will just be concentrating on those that get converted to pyruvate. How amino acids are degraded will be discussed under the topic of protein metabolism in the future. That is it for lactate and glucogenic amino acids. Glycerol is also gluconeogenic. It gets phosphorylated by glycerol kinase, which means that the phosphate group that was transferred to glycerol is from ATP. This yields glycerol 3-phosphate, which you hopefully might remember from the phosphoglycerol shuttle as mentioned in the oxidative phosphorylation video series. Glycerol 3-phosphate then encounters NAD+ dependent cytosolic glycerol 3-phosphate dehydrogenase and is oxidized by it to dihydroxyacetone phosphate or DAP. The utilization of propionate in the form of propionyl-CoA is of gluconeogenic significance only among ruminants. In humans, this is a very minor source of carbons for gluconeogenesis. Propionyl-CoA, by the way, is the result of oxidation of fatty acids with an odd number of carbon atoms. Now, such fatty acids are obtained mainly from vegetables in the diet. Propionate is also a major microbial fermentation metabolite in the large intestine of humans. Now, there is good evidence to suggest that propionate actually has nutritive value to the intestinal endothelium. Propionyl-CoA undergoes an ATP-dependent carboxylation reaction to form dimethylmalonyl-CoA. This is catalyzed by propionyl-CoA carboxylase. Dimethylmalonyl-CoA is then stereochemically inverted to L-methylmalonyl-CoA. This reaction is catalyzed by a vitamin B12-dependent enzyme called methylmalonyl-CoA racemase. L-methylmalonyl-CoA is then converted to succinyl-CoA in a reaction catalyzed by methylmalonyl-CoA mutase. Succinyl-CoA can then be converted to pyruvate and enter into gluconeogenesis. Gluconeogenesis is said to occur by a pathway that reverses many of the steps of glycolysis. As what you've hopefully learned from the glycolysis video series, many steps in the glycolytic pathway are reversible, though there are some that are highly exergonic and thus irreversible. Let's take a look back at the glycolytic pathway. This is a preparatory phase of glycolysis. Recall that the steps catalyzed by hexokinase or glucokinase and PFK1 use ATP, are overall exergonic, and thus are unidirectional and irreversible. This is the ATP generating or payoff phase of glycolysis. The steps that generate ATP are those catalyzed by phosphoglycerate kinase and pyruvate kinase. 2 ATPs for each molecule of glyceraldehyde 3-phosphate or GAP, and thus 4 for each molecule of glucose that enters glycolysis. Recall as well that the phosphoglycerate kinase step is the only reaction of glycolysis that involves ATP and yet is reversible under normal cell conditions. This makes only the pyruvate kinase step in the payoff phase the only irreversible step. Hence the statement from earlier, gluconeogenesis occurs by a pathway that reverses many, but not all, of the steps of glycolysis. For each exergonic step of glycolysis, an endergonic reaction must technically occur to reverse that step. And we already know that that is thermodynamically unfavorable. In order to understand gluconeogenesis, we have to know what happens in the opposite direction in those steps that are exergonic or irreversible in glycolysis. Let us divide the pathway of gluconeogenesis as such. Those parts in orange are steps that are simply the reversal of the corresponding steps in glycolysis, using the same enzymes. Those steps in green, on the other hand, correspond to the irreversible steps in glycolysis and require different enzymes in order for these steps to occur in the direction of gluconeogenesis. Let us look at the first step in gluconeogenesis, which corresponds to the last step of glycolysis. This step does not entail direct conversion of pyruvate to phosphoenol pyruvate or PEP. Pyruvate must first be converted to oxaloacetate, and this reaction involves incorporating a molecule of carbon dioxide to the 3-carbon chain, a process called carboxylation. This is catalyzed by pyruvate carboxylase, which requires a coenzyme biotin or vitamin B7. Since this step involves formation of covalent bonds, which is endergonic, ATP is required to drive this reaction forward. This reaction is often referred to as an anaplerotic reaction. Anaplerosis is defined as the act of replenishing Krebs cycle intermediates that have initially been extracted for biosynthesis. You will learn in future video series that the Krebs cycle is a hub of metabolism, with central importance in both energy production and biosynthesis. Next, oxaloacetate is simultaneously decarboxylated and phosphorylated to phosphoenol pyruvate or PEP. This reaction is catalyzed by phosphoenol pyruvate carboxykinase, which can be abbreviated as PEPCK. PEPCK is distributed about equally in both cytosol and mitochondria. The phosphate group is from GTP. In the liver and kidneys, where gluconeogenesis occurs, GTP is provided by the Krebs cycle, which you will learn in the near future. Such a setup provides a link between the Krebs cycle activity and gluconeogenesis in order to prevent removal of too much oxaloacetate for gluconeogenesis, which would in turn impair Krebs cycle activity. Now, this step is a rate-limiting step of gluconeogenesis. The formation of PEP is also an endergonic process. The splitting of GTP provides the free energy to drive this endergonic reaction forward. Recall that glycolysis occurs in the cytosol. So, pyruvate is produced in the cytosol as well. Its oxidation to acetyl-CoA, however, occurs in the mitochondrial matrix. So, it must be transported into the mitochondria by a pyruvate transporter. So, technically, pyruvate exists in both the cytosol and the mitochondrial matrix. Since gluconeogenesis occurs in the cytosol, cytosolic pyruvate is converted to oxaloacetate, and there will be no problem. But what about the oxaloacetate in the mitochondria? How does it enter the cytosol? Recall that the inner mitochondrial membrane is freely permeable only to oxygen, carbon dioxide, and water. It is much less permeable to ions and small molecules compared to the outer mitochondrial membrane. This way there is compartmentation, that is, separation of the matrix from the cytosolic environment. The only way in which oxaloacetate in the matrix is, in quotation marks, transported out of the mitochondria is by way of the malate or malate aspartate shuttle, which you've already learned in the previous video series. As a review, this shuttle translocates electrons produced during glycolysis across the inner mitochondrial membrane for oxidative phosphorylation. Simply refer to the glycolysis video series for more details about the malate shuttle. Now, that covers the first step in gluconeogenesis. The next chunk of the gluconeogenesis pathway we will look into is the conversion of PEP all the way to fructose 1,6-bisphosphate. The location of gluconeogenesis, as we all know, is in the cytosol. Starting with PEP, the steps of glycolysis are reversed to form glyceraldehyde 3-phosphate or GAP. For every 2 molecules of glyceraldehyde 3-phosphate that are formed, 1 is converted to dihydroxyacetone phosphate or DAP. Recall that DAP and GAP are triose phosphates. These 2 triose phosphates then condense via reversal of the aldolase reaction to form fructose 1,6-bisphosphate. So, long story short, this segment or chunk of the gluconeogenic pathway is simply the reversal of the corresponding steps in the glycolytic pathway. We are now down to this remaining part. A closer look at this portion of the pathway reveals that 2 of the 3 steps are not the simple reversal of the corresponding steps in glycolysis. Let's look at the step that converts fructose 1,6-bisphosphate back to fructose 6-phosphate. This step is not a reversal of the reaction involving PFK1. This step involves the removal of phosphate from fructose 1,6-bisphosphate without reformation of ATP. This process is catalyzed by a phosphatase called fructose bisphosphatase and not by a kinase such as PFK1. Clearly, this step in gluconeogenesis is not a mirror image of its corresponding step in glycolysis. Now, before going any further, review on your own the difference between kinases, phosphorylases, and phosphatases in terms of the kind of biochemical tasks they perform. Now, for the next and penultimate step in gluconeogenesis, there's really nothing much to say except that it is the reversal of the corresponding step in glycolysis. The last step in gluconeogenesis corresponds to the step in glycolysis catalyzed by hexokinase or glucokinase. It is similar to the step corresponding to the one catalyzed by PFK1 in that this step is not a reversal of the glucokinase or hexokinase reaction. This step involves the removal of phosphate from glucose 6-phosphate without reformation of ATP. Now, this process is catalyzed by a phosphatase called glucose 6-phosphatase and not by hexokinase or glucokinase. Of note, the enzyme glucose 6-phosphatase is present in the liver and kidney, but not in the muscle. This enzyme allows the liver to export glucose into the bloodstream. And that concludes the pathway of gluconeogenesis. From the previous video, we learned that gluconeogenesis occurs mainly in the liver under fasting conditions. There are actually other conditions wherein gluconeogenesis can take place, namely prolonged exercise, ingesting a high-protein diet, and during conditions of stress, which include critical illness. Now, whether or not glucose metabolism will go in the direction of gluconeogenesis will depend on the substrate that's available, as well as the activity or the amount of key regulatory enzymes that are present. Let's talk first about substrate availability regulating gluconeogenesis. Since gluconeogenesis occurs in the liver, flow of major substrates from peripheral tissues has to take place for its activity to increase. One of those substrates is glycerol. In times of fasting, when insulin levels decrease and levels of glucagon, epinephrine, and cortisol increase, glycerol is mobilized from the adipose tissue and it makes its way to the liver. During exercise, skeletal muscle produces lactate, which then goes to the liver to be converted to glucose via the Cori cycle, which will be discussed in the next video. During times of stress and critical illness, skeletal muscle undergoes catabolism, and the constituent amino acids are brought to the liver as substrates for gluconeogenesis. Now, besides substrate availability, I mentioned that gluconeogenic activity is also influenced by the amount and activity of regulatory enzymes. These enzymes would correspond to the steps in glycolysis that are catalyzed by regulatory enzymes, namely the step catalyzed by pyruvate kinase, phosphofructokinase 1, and hexokinase. Recall that pyruvate, which is the end product of glycolysis, is derived from lactate and amino acids, particularly alanine. Recall as well that under anaerobic conditions, pyruvate is converted back to lactic acid. Under aerobic conditions in the fed state, however, it is converted to acetyl-CoA by pyruvate dehydrogenase, and this eventually enters a Krebs cycle. Under conditions that favor gluconeogenesis, on the other hand, this happens. During gluconeogenesis, the pyruvate dehydrogenase that is supposed to convert pyruvate to acetyl-CoA in the fed state becomes inactive. Recall that conditions favoring gluconeogenesis occurs during the fasting state. During fasting, insulin levels decrease and glucagon levels increase. The increased glucagon levels appear to have a minor effect in mobilizing fatty acids and glycerol from adipose tissue. More importantly, though, glucagon stimulates fatty acid beta oxidation by inhibiting synthesis of acetyl-CoA carboxylase, something that you will learn eventually under fatty acid biosynthesis. With beta oxidation of fatty acids, the byproducts acetyl-CoA, NADH, and ATP increase in amounts. Concurrently, ADP levels decrease. Now, these high levels of acetyl-CoA, NADH, and ATP, and the concurrent nutrient deprivation that is consequent of the fasting state, cause activation of pyruvate dehydrogenase kinases, or PDKs. These kinases attach inhibitory phosphates on the pyruvate dehydrogenase complex, thus explaining why this enzyme is inactive during gluconeogenesis. In addition to pyruvate dehydrogenase becoming inactive during gluconeogenesis, pyruvate carboxylase, which catalyzes the conversion of pyruvate to oxaloacetate, becomes active. This is due to its allostatic activation by acetyl-CoA, which is produced during fatty acid beta oxidation that occurs during fasting. Thus, acetyl-CoA reciprocally regulates pyruvate dehydrogenase and pyruvate carboxylase. Besides inhibition of pyruvate dehydrogenase and activation of pyruvate carboxylase, PepCK is induced. So PepCK is what's called an inducible enzyme. An enzyme is said to be inducible if its quantity increases because of increased transcription of its gene and increased translation of its mRNA. What then would increase expression of the PepCK gene? We already know that glucagon and epinephrine are gluconeogenic hormones. They stimulate G protein-coupled receptors on hepatocytes. This leads to a cascade of events that lead to activation of an effector enzyme called adenylyl cyclase or adenylate cyclase, which in turn produces the second messenger cyclic AMP, which in turn activates a kinase called protein kinase A or pKa. The catalytic subunit of pKa makes its way in the nucleus where it phosphorylates and activates cAMP response element binding protein or CREB. CREB in turn functions as a transcription factor that enhances transcription and expression of the PepCK gene. Of note, cortisol, another stress hormone, also induces PepCK, but through a different regulatory site on the PepCK promoter. Let's have a recap now. The reasons why the conversion of pyruvate to oxaloacetate, then to phosphoenol pyruvate, is favored during gluconeogenesis is because of these three events. There's still a fourth one though. It's the inactivation of pyruvate kinase. Think about it. If pyruvate kinase was kept active, all the phosphoenol pyruvate that you've been synthesizing under gluconeogenesis will just get reconverted back to pyruvate. These substrates would simply cycle, causing a net loss of energy with no net generation of useful products. And this would defeat the whole purpose of performing gluconeogenesis. How do we inactivate pyruvate kinase then? Recall from earlier that glycogen, whose levels are high during the fasting state, activate protein kinase A via the adenylyl cyclase pathway. The activated pKa would then phosphorylate and inactivate pyruvate kinase, effectively preventing the reconversion of phosphoenol pyruvate back to pyruvate. Recall that this step in gluconeogenesis is not the simple reversal of the same step in glycolysis. The fructose 1,6-bisphosphate to fructose 6-phosphate reaction is catalyzed by fructose 1,6-bisphosphatase, while the reverse is catalyzed by phosphofructokinase 1, an important regulatory enzyme in the glycolytic pathway. To understand regulation at this particular step in gluconeogenesis, we need to be familiar with another important regulatory enzyme called phosphofructokinase 2 or PFK2. Phosphofructokinase 2 or PFK2 is a bifunctional enzyme that has both kinase activity and phosphatase activity. The kinase function of this enzyme effects the addition of a phosphate group to the carbon 2 position of fructose 6-phosphate, while the phosphatase part, which is fructose 2,6-bisphosphatase, removes this same phosphate group. For that matter, PFK2 is under allostatic control of fructose 6-phosphate. As levels of fructose 6-phosphate increases, the kinase activity is activated while the fructose 2,6-bisphosphatase activity is inhibited. The abundance of glucose, which occurs during the fed state, as you already know, encourages the rise in fructose 6-phosphate levels via the glycolytic pathway. As mentioned just previously, with this rise in fructose 6-phosphate levels, the kinase activity of PFK2 is activated in turn, while its phosphatase activity is inhibited. In effect, fructose 2,6-bisphosphate is produced. Eventually, levels of fructose 2,6-bisphosphate increase. These increased levels allosterically activate PFK1 of the glycolytic pathway. As a result, glycolysis ensues. Now, in addition to allosterically activating PFK1, increased levels of fructose 2,6-bisphosphate also inhibit fructose 1,6-bisphosphatase in the opposite direction. Now, fructose 6-phosphate from earlier also prevents the activation of the phosphatase function of PFK2. Now, over time, however, as glucose supplies dwindle as the individual progresses more into the fasting state, glucagon levels then increase, leading to the activation of the denylate cyclase pathway. Now, with decreasing levels of fructose 6-phosphate, the kinase activity of PFK2 then becomes inhibited, and the fructose 2,6-bisphosphatase activity gets activated. Now, this promotes reconversion of fructose 2,6-bisphosphate back to fructose 6-phosphate, consequently causing a drop in fructose 2,6-bisphosphate levels. This removes the allostatic activation of PFK1 while simultaneously removing the allostatic inhibition on fructose 1,6-bisphosphatase. Now, additionally, the synthesis of fructose 1,6-bisphosphatase is also induced during fasting. As a result of all this, gluconeogenesis ensues. Now, just for completion, protein kinase A has a functional opposite, protein phosphatase 2. This removes a phosphate on PFK2 initially attached by protein kinase A, effectively activating the PFK2 kinase function and inactivating the PFK2 phosphatase function. This occurs during the fed state, when the individual has ingested a meal following the fasting state. Now, in the fed state, the whole process starts all over again. Just to mention again, besides the cascade of activities that was just extensively discussed, the synthesis of fructose 1,6-bisphosphatase is also induced during fasting, promoting the reaction that you see as heading of this particular slide. Now, as you learned previously, this glucose 6-phosphate to glucose step is not a reversal of the reaction catalyzed by glucokinase or hexokinase. What catalyzes the reverse reaction then? Definitely not glucokinase as discussed earlier. Also, this enzyme is relatively inactive during gluconeogenesis. Instead, the enzyme involved is glucose 6-phosphatase, which cleaves off the phosphate group from the carbon-6 position of glucose-6-phosphate. This particular gluconeogenic step does not produce ATP, since the phosphate bond of glucose-6-phosphate is of low energy. Now, let's try to apply what you've learned under enzymology. What do you remember about the kinetics of glucokinase? You can pause this video for a while to think about what I just asked before I reveal the answer. Now, recall that in most tissues, except the liver and pancreatic beta cells, hexokinase catalyzes the glucose to glucose-6-phosphate step. Hexokinase has a very low Km, and as a consequence, only a very small concentration of its substrate, glucose, is necessary for the enzyme to reach maximum velocity or Vmax. Now, once the product, glucose-6-phosphate, accumulates intracellularly, an allosteric product inhibition on hexokinase occurs, thereby halting glucose internalization and utilization by cells. In tissues that use hexokinase, however, there exists no enzyme intended to remove the phosphate covalently attached to the carbon-6 position of glucose. Now, in liver and pancreatic beta cells, an isoform of hexokinase, glucokinase, is used. It has a very high Km, and because of this, an incredibly high blood glucose level must first be obtained in order to saturate the enzyme and reach Vmax. Unlike with hexokinase, though, the glucose-6-phosphate that accumulates intracellularly does not exert any allosteric product inhibition on glucokinase, because for one thing, it is eventually incorporated into the growing molecule of glycogen. Now, for that matter, with the induction of the hormone insulin, the liver continues on internalizing glucose to form glycogen until a point where blood glucose levels dictate that insulin withdraws its activity. Glycolysis can continue in the liver even when energy levels are high, so that anabolic pathways, such as synthesis of major energy storage compounds, glycogen and fatty acids, can occur. As for reversing this step in glycolysis, the liver possesses an enzyme called glucose-6-phosphatase, which removes the phosphate group on the carbon-6 position of glucose, initially added by glucokinase. Its activity increases during fasting to allow glycogen stores to be broken down into its glucose constituents for utilization in basic metabolism. Moreover, glucokinase becomes inactive during fasting because a drop in blood glucose means moving further and further away from its Km of 5 to 6 millimolars. This figure shows the kinetics of hexokinase and glucokinase. Owing to the very low Km of hexokinase, it is able to quickly reach Vmax with just a small concentration of its substrate, glucose. Glucokinase, on the other hand, requires a higher concentration of glucose just to get to half Vmax and an even higher concentration to reach Vmax. Now, here's a table that summarizes the activities of various enzymes in glucose metabolism in response to insulin and glucagon. The covalent modification involved here is mostly phosphorylation. The conformational change afforded by this type of covalent modification makes certain enzymes more active and other enzymes less active. Take time to go through this table. The enzymes glycogen synthase, phosphorylase kinase, and glycogen phosphorylase will be tackled under glycogen metabolism. Here's a recap of what was covered so far. We derived glucose from digestible dietary carbohydrates, with galactose and fructose readily converted to glucose in the liver. Glucose is also formed from basically two groups of compounds that undergo gluconeogenesis, namely amino acids, glycerol, and to a lesser extent propionate, which leads to net conversion to glucose, and of course lactate from skeletal muscles and RBCs. But what happens to lactate when it is transported to the liver? It is converted to pyruvate, adding to the pool contributed by the usual glycolytic activity. Skeletal muscles supply enough pyruvate that is shuttled to the liver. A reversal of the glycolytic pathway then ensues in the liver, where the shuttled pyruvate gets converted back to glucose-6-phosphate. Through the action of glucose-6-phosphate, which is found only in the liver, glucose gets liberated and returned to the bloodstream, where it finds its way back to the skeletal muscles for utilization. Now, this cycling of lactate by the liver back to the peripheral tissues in the form of glucose comprises the Cori cycle or the lactic acid cycle. Now, conversion of lactate back to glucose is in itself an energy demanding process, as evidenced by the requirement of 6 molecules of ATP, which does not match the net of 2 ATP molecules produced by glycolysis. Now, fortunately, the liver is also the site of fatty acid beta oxidation, and ATP molecules derived from it can be used to power gluconeogenesis. Alternatively, when muscles degrade amino acids for energy needs, for example during fasting or starvation, the resulting nitrogen is transaminated via alanine transaminase to pyruvate to form the amino acid alanine. Alanine is then exported to the liver, where it is again transaminated back to pyruvate. From there, pyruvate becomes a substrate for gluconeogenesis, and the resultant glucose is then returned back into the bloodstream for utilization. Now, this cycling of the amino groups and carbons of alanine by the liver back to the peripheral tissues in the form of glucose comprises the Cahill or the glucose-alanine cycle. Now, this figure shows the Cahill cycle laid alongside the Cori cycle. Just as with the Cori cycle, the conversion of pyruvate back to glucose demands the use of 6 molecules of ATP per molecule of glucose. Fortunately, ATP required for the hepatic synthesis of glucose from pyruvate is derived from the oxidation of fatty acids.
Thank you, doctors. Thank you, nurses. Thanks for working.